Hello everyone, and welcome back to Big A's Biology Funganza, the sequel. In today's episode, we're talking all about the miracles of RNA and protein synthesis. So you may be wondering, how does protein synthesis work? How do we make these proteins that basically run our entire body? Well, that's exactly what you've come here to find out today. So there's basically two main steps in the process of protein synthesis. The first one is transcription, and the second one, translation. DNA is basically the blueprints for all sorts of different things in your body. So if you look at this DNA here, you'll see that it's a double helix shape made up of two parts. You got the backbone and you got the rung. The backbone's made up of two different molecules, a phosphate group and a deoxyribose sugar. And the rung is made up of two nitrogenous bases. In DNA, the nitrogenous bases are thymine, guanine, adenine, and cytosine. There's also four different types of nitrogenous bases for RNA too: guanine, adenine, cytosine, and uracil instead of thymine. Each nitrogenous base is connected using a hydrogen bond to its complementary base. In DNA, adenine always bonds with thymine, guanine bonds with cytosine, and in RNA, guanine still bonds with cytosine, and adenine bonds with uracil.
A gene is a section of DNA that codes for a specific protein, and these are what we'll use to make our protein. DNA holds all the hereditary material that gets passed down in variety from generation to generation. Keratin is a protein in the body that helps form hair, nails, and the outer layer of skin in the body. If you don't eat enough keratin or your body doesn't produce enough, then your hair will grow at a slower rate while becoming more brittle and less shiny. Keratin is just one example of a protein that plays an important role in the human body. Some proteins are used in transportation, some are used as structure, as enzymes, in protecting the body, and that's just a few of the many reasons why we need proteins. Let's talk about how proteins are made, aka protein synthesis. Transcription is when DNA gets transcribed into a message. Transcription happens in the nucleus in eukaryotic cells, and in prokaryotic cells it happens in the cytoplasm. Enzymes are the key to all of protein synthesis happening. RNA polymerase approaches the DNA molecule as it gets quote-unquote unzipped by DNA helicase. This process is known as initiation, which is the beginning of transcription, and the RNA polymerase can start its job. An RNA polymerase enzyme goes to both of the recently unzipped DNA strands and uses it as a template strand to make mRNA, aka messenger RNA. The RNA polymerase connects complementary RNA bases to the DNA strand. The base pairing is as follows. Instead of adenine bonding with thymine, like it does in DNA, the adenine bonds with uracil because there is no thymine in RNA. As always, the guanine still bonds with the cytosine. As the mRNA is growing down the 3' side of the DNA strand, this is known as elongation, and when this process finishes, it is known as termination. There can be mutations in this process when the RNA polymerase copies it in the wrong way. After this whole process is finished, a single mRNA strand is made. There are two parts of the DNA strand: the exon and the intron. The exon parts can be used to code, while the introns cannot be used to code. The intron parts of the DNA get removed in a process called RNA splicing. After that, the mRNA is finished and it goes through the nuclear pore and into the cytoplasm.
But guess what? After all that's we're only halfway there, man. We still have so long to go. But anyway, so now we got the mRNA. That thing has to somehow turn into a polypeptide. Well, what happens is the mRNA floats around until it finds a ribosome. The ribosome will latch onto the mRNA and find the initiation point, which is actually a 3-letter sequence or codon on the RNA that dictates the start of synthesis. This sequence is always AUG, and it codes for the amino acid methionine. Methionine is always going to be the first amino acid in a chain, but you know, how does it get to the right place at the right time? I mean, that's a lot of coordination, right? Well, this is with the help of tRNA. There's a 3-letter sequence on each tRNA called an anticodon, which links to the complementary codon on the mRNA. Each tRNA has an amino acid on it, so one tRNA goes to the A site of the ribosome, attaches its amino acid to the chain, and then it holds the entire chain in the P site until the next one comes along, and the chain is built in the process of elongation. Eventually, the ribosome is going to have to stop building this protein, and that is with the help of a stop codon, which is the termination point. The finished polypeptide chain is going to be released into the cytoplasm, and it'll assemble into its final protein shape all by itself. And this is because the polarity of amino acids will fold the protein in the way it's meant to be folded, and hence a protein is born. Also, the polarity of amino acids is the reason mutations can mess up the shape of a protein, because if you have a different amino acid, it's going to have different polarity, and it's going to just fold completely different, and it can denature it. Well, anyway, thanks for coming to Big A's Biology Funganza, the sequel featuring Lil D. And I hope you had a great time. I hope you learned something. Also, please give us a good grade. Bye!